Welcome to the biology department. I guess first I'll tell you a little bit about myself. I got my bachelor's and master's degree from Midwestern, from here. I'm actually a native of Wichita Falls, born and raised here. And then I did my PhD in entomology at Texas A&M University down in College Station. So that's my education background. I'm working together with Drew Skinner, one of your classmates here, and we're working together on this project with Aquatic Insects that are coming out of the Wichita River, which is a river that flows right through town here. But the reaches of the river that we're working with are actually about 3 counties over from here, the majority of the sites that these aquatic insects are sampled from. So they're a pretty good ways upstream of the river. And one of the key ingredients that eventually we hope to look at in this whole project is that along that river, because at some of the origins of places where the water flows into this river, there's a lot of salted land. So the end result is that there's varying grades of salinity within that river as you come down the lengths of that river. So one of the things we hope to address eventually is there an effect of the differences in salinity in that water on the types of insects, aquatic insects that are living within that water. Come on in, have a seat. So that's eventually what we hope to do. But right now, the main thing we're looking at, trying to sort out, is what's there. What insects are there? So this originally started as a project where I had one of my graduate students that was working on it, that worked for the USGS, U.S. Geological Survey. And she had access to these sites where these specimens were collected. Well, unfortunately, USGS had a cutback at the time. She was one of the latest people to be hired, so she was one of the first people to be let go. And therefore, she was no longer to continue on that project and ended up having to switch to a different project. But prior to that happening, she had already collected a pretty good amount of data. So that being specimens, aquatic specimens, that I've had in my lab since then. And this was done, she was working on that at the end of 2010 into early 2011. So just this last week, Drew and I got through sorting of the samples, macro sorting, which is basically getting a general idea of what we're working with and what kind of volume we're talking about. And the number of specimens we're working with is a little bit over 1,900 individual specimens. So we got a decent sample size of what we're looking at, even though it was only over a course of about 6 months, I think. So anyway, it's kind of a spin-off of what we're actually doing in the lab. What I decided to do for this workshop with y'all is, first of all, introduce you to one of the tools that we use in this job we're doing, and to some of the principles of microscopy. So we're going to be looking at using a microscope today, and a lot of times, even if you're not in the sciences, usually students at some point in time have used a microscope. But even though you have maybe used a microscope in the past, you maybe haven't used that microscope to its full potential because you weren't instructed properly on all the aspects of using that tool. Because essentially what it's meant to be is an extension of your senses, of your sight, to extend your sight to be able to look at things at a much larger magnification. Also, with that being said, typically most of the scopes that students might have used in junior high or high school or what have you are usually compound microscopes, which are like these microscopes here, where you've got a nose piece here that has rotating objectives, and you've got a stage up here where you look at slides, and look at specimens on slides. Well, that's not what we're going to be using today. What we're going to be using today is what we call a dissection scope, which is like this, which doesn't give as much power of magnification as what you can get with a compound microscope. But the advantage of this, for what we're doing, is that this allows you to look at things that are a little bit larger in size, something that isn't so small necessarily that you would have to mount it on a microscopic slide. So you've got a big stage area here to be able to work with the specimens. Okay, so that's the first part of this workshop that I want us to do, is to work with these dissection microscopes. These are fairly new scopes we have. They're Zeiss microscopes, which if you're familiar with microscopy at all, Zeiss is one of the leading companies there is. I mean, you go to major research hospitals and so forth, and you look at what kind of optics that they use, what kind of optical equipment, oftentimes it's going to be Zeiss, because of the high quality that the lenses provide for you that are in these particular pieces of equipment. So really good scopes. So I want each of you to get one of these microscopes to look at. And first thing you need to do is make sure that you carry it correctly. These particular models, DV4s, they have actually a carrying handle up here at the top. So you pick it up, and you also support it from underneath. And it's also a good practice not to jostle these around when you're carrying them, especially when you set them down. You set them down very gently so that you don't jar around the lenses which are internal to the body of these scopes. Okay, so I want each of you to grab one of these microscopes, but also I want you to grab a power cord that goes with it. Those power cords, the scopes, if you look back here, are in these middle two cabinets. These are the scopes you need to choose from. And the power cords you're going to get are in this box right here, this bigger box. Grab one of those, grab your scope, grab your power source, and bring it back to the table.
She's like one thing out there. There's like one more on March 25th. I just want to get out of the way. She's not living here. We asked her about a couple days ago. She said,
Another thing you'll notice is you bring these scopes and set them down. So on the bottom of the microscopes, they actually have rubberized feet. Those feet are rubberized for a purpose, and that is to prevent the microscope from being able to slide on the table. So you should never slide the microscope on the table. You need to turn it around or what have you, and pick it up and reposition it, because sometimes that sliding, if it catches, causes jarring of the microscope, which again can damage the alignment of the optics inside of that. That's okay. Go ahead. Position it to where the eyepiece will be facing toward you. And then what we're going to do, we're going to go through the use of this. I'm going to use this scope up here and point out things that you need to do as you get prepared to use this tool. We'll be using it in a little project. Okay, so everybody's got a scope, everybody has a light source. Next thing you need to do is take that cord that you've got and plug it in over here at the base on the right-hand side. That plug, the end of that should fit in there, and then plug the other part into the electrical socket. And then over on the right-hand side at the base of the scope, you'll see a big capital M there. If you push on that, that's what turns on the first light source. Just push it one time. It's a touch-sensitive control. Okay, everybody kind of got to that point. Now, let me go ahead and mention another thing here before we get further along about lighting. With microscopy, we basically use one of two types of lighting. If you want to look at something like on the outside, the outer surface of something, you use reflected light. That is light being shown down on the object where it reflects back so that you can see that image. That's what you have in play right now. Now bash that big M button again the second time. Now notice the light is coming out of the baseline. This light source is transmitted light. This is meant to be a light source that is going to shine through a specimen and illuminate something that, say, may be transparent. Say you want to look at the internal parts of, say, a certain little microscopic organism. You may have it mounted on the slide. Put that on the stage over this source, it shines through it, and you're using transmitted light instead of reflective light to illuminate the specimen that you're going to be looking at under the microscope. Now, if you push that button, the M, one more time, you'll now have both of those light sources in play, both transmitted and reflected light. Now, you'll also notice on either side of the M there, you'll see there's a plus and a minus. If you push those, if you push plus, you ought to see that the light sources start getting brighter. If you push the minus, you see it's not as bright. So those are the buttons that are used to change the light intensity, the amount of intensity of light that is being used with your specimen. Now if you push that M button one more time, it shuts all the light sources off. Okay? So go ahead and push it one more time and turn on just the reflected light, because that's what we're going to be using today. Okay? Another feature of these microscopes, when you're using reflected light, one of the major things that comes into play is oftentimes the angle of the light. The angle that the light is coming in on your specimen. And a lot of times the angling of light is used to highlight and illuminate, say, certain parts of a specimen that you're trying to look at. And I show this to students all the time in classes when I'm teaching labs. There are instances where you can have light shining on a specimen and you're looking for a particular feature. And because of the angle of the light, you could swear there isn't this particular feature there, like maybe a hair on one part of the body. You're trying to see if there's a presence of this hair or bristle there or not. But all you have to do is just change the angle of light ever so little, and all of a sudden it's like it appears before your eyes. It all had to do with the angle of the light. So the angle of the light is important. And if you notice underneath the head of the scope here, see those little blue paddle-like structures? Those are used to change the angle of the light a little bit with these microscopes. Now, ideally, what we usually do, because for most things that we do with entomology and so forth, that's usually not enough of a change of an angle of light. And in fact, the reflected light sources that you have in these scopes usually aren't sufficient for more of the projects that we do, like what Drew and I are working on. So instead, then what we do is we bring in the big guns. We bring in an additional light source. This light source that you're looking at here, this is what's called a dual gooseneck. This is a fiber optic light source. So you're able to actually position from all different angles, because you can twist and turn these goosenecks however you want, bringing in light from both sides if you want, or you can eliminate it from one side. You can change the light intensity here. This is cold light that shines in on your specimen and does an awesome job of illuminating with reflective light. And this light, the intensity can be so bright that, I mean, you got to watch. I mean, if you go and look and you have it too bright, it's like it really, you can tell the effect on your retinas in your eyes. So it's something you got to kind of be careful with sometimes as well. Okay, so enough about light. Now, the next thing you should understand about use of a microscope, not only of this type, but also compound microscopes. These being an extension of your senses, these things are made for them to be manipulated and adjusted for you personally. for your body, the way your body is set up. For instance, the distance of one eye to the other. What we call the interocular distance. That's adjustable. Just like if you were to use a pair of binoculars. If somebody hands you a pair